Hello everyone. Today's topic is deoxyribonucleic acid DNA. I will discuss this topic in details. If you like this video, please like, comment and subscribe my channel and also share it with your friends. First, DNA. DNA or deoxyribonucleic acid is a complex structure hereditary materials in humans and all other organisms that carry and transfer genetic information from parents to offspring. So DNA, that stands for deoxyribonucleic acid. It is a complex structure and hereditary materials that helps in the human and all other organisms to carry and transfer genetic information from parents to offspring or from one generation to another generation. You can see that this is a structure of a cell where there is a nucleus, mitochondria, centrosome, ER means endoplasmic reticulum. This is the smooth endoplasmic reticulum. Then rough endoplasmic reticulum, ribosome that helps protein synthesis. PROTEIN, protein synthesis. Then cytoplasm. Then in this nucleus, this is also a picture of nucleus, and in the middle there is a nucleolus, and this is a chromatin. And this chromatin is also form a chromosome when in cell division during prophase stage. The chromatin is converted to chromosome, and in the chromosome that this is all our DNA. It is a short arm, and this is a long arm, and in the middle it is called centromere. Then coming to the next point, that is the location of DNA. DNA is found in all prokaryotic and eukaryotic cell. It is mainly located inside the cell nucleus, that is called nuclear DNA, and a small amount of DNA also present in mitochondria. So that's called mitochondrial DNA or mtDNA. DNA are mainly present in two places. One is nucleus and another is mitochondria. Then who discovered the DNA? It was discovered by in 1869. Johann Friedrich Micher first recognized and discovered DNA molecules. In 1952, Rosalind Frank used X-ray crystallography to visualize the helical structure of DNA. In the 1953, after 1 year, James D. Watson and Francis Crick discovered the helical structure of DNA molecules. Then the structure of DNA. DNA structure. It is a right-handed double helix structure. There are about 10 nucleotide pair per helical turn. Then the DNA has three main components: deoxyribose, a pentose sugar, where pento means 5 and tose means sugar. Then two nitrogenous base: adenine, guanine, thymine, cytosine, and the last phosphate. This is the structure of deoxyribose sugar, and it is a 2-deoxyribose sugar because it is 1 number of carbon, 2 number of carbon, where there is an only H group. Deoxy means removal of O group. So there is a removal of O group, so it's called deoxyribose because the ribose structure is almost same. OH, H, OH, H, OH, and CH2OH and H, but there it is present OH group. But in the deoxyribose sugar there is only H molecules. O is removed, so it's called deoxyribose. Then the nitrogenous base, purine and pyrimidine, two nitrogenous bases are present. Purine is made of 6-member ring and fused to a 5-member ring. You can see that this is the 6-member ring. This is the structure of adenine and this is the structure of guanine. You can see this picture and this picture, both are almost same, but there is an double bonded O and there is a NH2 groups, and here this is an NH group but there is no NH2 groups. This is the only difference between adenine and guanine. And then the pyrimidine. Pyrimidine is made of 6-member ring. There is only 6-member ring, you can see, but there is also a 5-member ring present, but in this structure there is no 5-member ring present. So this is a single ring structure. Yeah, you can see that this is a thymine structure, this is a cytosine structure, and this is a uracil structure, where in the thymine and in the uracil there is a double bonded O groups, but in the cytosine there is an NH2 groups, and all the structure almost same. In case of the thymine there is an group CH3, but there is no CH3, but in case of DNA there is present only thymine and cytosine, not uracil. Uracil is present in DNA, uracil is present in RNA, not DNA. Uracil is absent in DNA. Then the phosphate group. This is the structure of phosphoric acid that helps to make a bond between two nucleotides. You can see that this is an P phosphate and double bonded O. There is an single bonded OH and O negative that can easily bind with hydrogen ion due to this hydrogen bond is positive charge. Then I will discuss the DNA structure. In 1953, Watson and Crick described a 3-dimensional model of DNA structure from Franklin X-ray. Franklin is also a scientist who first see the structure of DNA with the help of the X-ray. So Watson and Crick first described the DNA structure with the help of the Franklin X-ray. Then first it is consists of two helical polynucleotide chain wound around each other in right-handed double helical structure. You can see that this is a double helical structure, one strand, and this is a second strand, this is a second strand, and this is a first strand. So it is a two helical structure and they around each other. Those are right-handed structure, means right-handed structure. In the helix, the two chain or the strand are anti-parallel and they run in opposite direction. You can see that this is the 5' end and this is the 3' end, and this is 5' and 3' end, so they are opposite to each other. Then in the third point, the sugar and phosphate chain are remains outside, means in this structure there is a sugar and there is a phosphate groups, where the bases adenine, guanine, thymine, cytosine all are present inside the structure. So the sugar and phosphate chain remains outside, so it's called the backbone of the sugar molecules, and purine and pyrimidine bases are remains on the inner side of the helix facing each other. This is the two chain that attach together by the hydrogen bond. The helix has a diameter of about 20 angstrom. The base pair are 0.34 nanometer apart in DNA helix. A complete turn of helix takes 3.4 nanometer. Therefore, in each helical turn, 10 base pairs are present. The external diameter of helix is 2 nanometer. You can see in this structure there is the small distance between this helical strand and this strand, so it's called minor groove. It is almost 12 angstrom, and the distance between this strand and this strand is the length between this strand and this strand is long, and so this is called a major groove. It is almost 22 angstrom. Then the distance between 5 prime to 3 prime, it is 2 nanometer, or it also called 20 angstrom. And this is a base pair and this is a base pair, so the distance between two base pair between two bases are called base pair, so it is about 0.34 nanometer. And the distance between a complete helix from here to here is 3.4 nanometer. And there is from this helix to this helix there is almost 10 base pair are present. So this is the overall structure of DNA molecules. In DNA molecules, two strands are joined each other by a phosphodiester bridge, which links a sugar of one nucleotide to a sugar of adjacent nucleotide. So one nucleotide is attached with its adjacent nucleotide by a phosphodiester bridge. Then the helix has two external grooves. The narrow grooves is called minor grooves. It helps the binding of small molecules, and while the wide grooves is called major grooves, it is helps in the DNA binding protein. Then there is a 4 nitrogen base mainly present: adenine, guanine, thymine and cytosine. Where adenine attached with thymine by 2 hydrogen bond and guanine attached with the cytosine by 3 hydrogen bond. Possible base pairs are A for adenine, T for thymine, G for guanine, C for cytosine. So adenine can bind with the thymine by 2 hydrogen bond or double bond, and same thymine is also bind with the adenine by 2 hydrogen bond. Guanine is bind with the cytosine by 3 hydrogen bond, and cytosine is bind with the guanine though a 3 hydrogen bond. So between adenine and thymine there is a double bond, and between guanine and cytosine there is a triple bond. The sequence of the bases are present on one strand of DNA molecules. A complementary sequence is present on the other strand of DNA molecules. Means if it is a first chain, so there is present adenine, thymine, adenine, cytosine, guanine, cytosine, adenine, cytosine. So in the opposite structure there is present adenine, so there is present thymine, so there is present adenine, thymine, cytosine. So in the opposite side there is present guanine, and in the guanine there is opposite present cytosine. As the same way, so adenine is also bind thymine and guanine is also bind cytosine, but adenine cannot bind with guanine or cytosine, or thymine cannot bind with guanine or cytosine. As the same, guanine cannot bind with adenine or thymine, or cytosine cannot bind with adenine or thymine. So this is also a possible base pair, only a base pair that is possible in DNA. If this is the first chain, so if the DNA sequence is like this, or nitrogen base is present like this, the second strand or the complementary strand, it is must that DNA sequence, possible sequence is this, like adenine, and in this opposite side there is present thymine, opposite side adenine, thymine, cytosine, guanine, cytosine, guanine, adenine, thymine, guanine, cytosine guanine. So this is the structure that helps you to understand DNA structure properly, that the phosphate group and sugar that is outside the DNA molecules, and inside there is a bases are present, adenine, thymine. Inside the structure there is present nitrogenous base, and they are formed bond with each other. They form hydrogen bond between each other. So this is a sugar molecules, this is a sugar molecules, and this is a phosphate groups, A for adenine, T for thymine, G for guanine, C for cytosine. So you can see that one phosphate group is attached with another sugar molecules, one sugar molecules with another sugar molecules, and between the structure there is a adenine can form a double bond between thymine, and as the same way in the other side there is a sugar molecules, phosphate molecules, and one sugar molecules is attached with another sugar molecules by a phosphate groups. Same, the second structure is almost same, only the bases are different from each other, that in this bases there is a thymine, and in this structure there is a adenine, which bond double bond each other and form a structure. Then the same, these sugar molecules and these sugar molecules are attached by the phosphate group, and this is also same, and this is also same, and where the cytosine is bond with the guanine by the triple bond, and guanine is bond with the cytosine by triple bond. So adenine guanine can form double bond, and cytosine guanine adenine guanine adenine thymine can form a bond, double bond, and guanine and cytosine form triple bond. So the chemical structure of DNA. Base pairing between adenine thymine, they form double bond. So this is the phosphoric acid or phosphate group, which is attached sugar molecules. This is a deoxyribose sugar molecules, because in this there is an only hydrogen bond, and in this structure there is 1 2 3. So in the 3 carbon there is an free OH group, where phosphate group is attached with the sugar. Then the sugar molecules is attached by C1N1 with the thymine. So this is the structure of thymine. And same, that there is on phosphate groups, and this is a deoxyribose sugar. It is also a present C3 3 number of carbon with the sugar to phosphate groups, and it is one number of carbon that is attached 1 2 3 4 5 6 7 8 9 N9 number of nitrogen in a adenine basis. So there is an free H groups that is bond with O, and NH groups that is bond with this NH. So they form a double bond. This is a one bond, and this is one bond. So there is a two hydrogen bond. They can form and form a DNA structure. As the same way, thymine and adenine are bond with each other. Then the base pair between cytosine and guanine. In this structure, that as the same, that this is the phosphate group. This is the phosphate group. This is the deoxyribose sugar molecules, and in the C3 the phosphate group is bind with the sugar molecules 1 2 3 4 and 5, and in the C3 and there is a N1. So C1 of the sugar molecules can bond with the N1 of the cytosine molecules, because this is the pyrimidine structure. Pyrimidine This is a pyrimidine structure. So in the pyrimidine structure, the N1 can bind with the C1. So this is the cytosine structure, and as the same that there is a phosphate group in C3 and C1. This is 1 2 3 4 5 6 7 8 9 8 9 of the guanine molecules can bind with the C1 of the sugar molecules. And in this structure there is an O groups that is attached with there is NH2 groups, that is where there is an hydrogen bond and oxygen are bond with each other. There is also a form a bond, and there is the same bond between hydrogen and oxygen. So there is 1 2 3 bond, 3 hydrogen bond is present. So cytosine and guanine can form 3 hydrogen bond. As the same way, guanine and cytosine can form 3 hydrogen bond. I think this structure between cytosine and cytosine and guanine and adenine and thymine, how they can bond each other and their chemical structure, I think that is clear for you. Then this is a chart where first is bases and sugar, and then deoxyribonucleoside, and if deoxyribonucleoside attached with the phosphate groups, now what is the name of deoxyribonucleotide? So when adenine and deoxyribose is bond with each other, they form deoxyadenosine, or it's called de-adenosine. When the phosphate group is attached with the deoxyadenosine, it is called deoxyadenosine monophosphate, DAMP, or D-adenyl acid, adenylic acid. Then guanine, when guanine is attached with the deoxyribose sugar, they form D or deoxyguanosine, and when the phosphate group is attached with the guanosine, it is called D-guanosine monophosphate or D-guanylic acid. When the cytosine and deoxyribose sugar are joined with each other, they form D-cytidine, and when with the D-cytidine or deoxycytidine is attached with the phosphate molecules, they form cytosine monophosphate, cytosine monophosphate, or D-cytidylic acid. Then as same, when the thiamine is attached with the ribose sugar, deoxyribose sugar, it is called D-thymidine. When the phosphate group is attached with the deoxythymidine, they form D-thymidine monophosphate or D-thymidylic acid. So there is almost same, G for guanosine and M for monophosphate, MP for monophosphate, C for cytidine, MP for monophosphate, and T for thymidine and MP for monophosphate. Then in the DNA, this is the most important important concept, that the chargaff rule. In 1940, Erwin Chargaff quantitatively analyzed DNA. He discovered that the number of nitrogenous base in the DNA was present in equal quantities, means where the amount of adenine is equal to the thymine and the amount of cytosine is equal to guanine. So DNA should have 1:1 ratio of purine and pyrimidine bases, means where adenine and guanine are present in same amount, and adenine and thymine are present in same amount, where the guanine and cytosine are present in same amount. So adenine and guanine, this is two are purine bases, and thymine and cytosine, this is a pyrimidine bases. So the adenine and guanine and cytosine and thymine is 1:1 ratio. So the equal adenine and thymine is present in equal number, and guanine and cytosine is present in equal number. But adenine and guanine cannot present in same number, or adenine or cytosine, they are not same number, or thymine or guanine or thymine or cytosine, they are also not present in same number. because they form a double bonded structure in DNA. DNA is a double bonded structure. So where is adenine group, it must be added adenine with the thymine. They cannot bind with the guanine or cytosine. So it is proven that adenine and cytosine is present in equal number. As the same way, guanine and cytosine is also present in same number. So the last point, double stranded DNA and RNA obey this rule. When they are formed double stranded DNA or RNA are formed double stranded, some are RNA, they are double stranded. So they they obey this rule, chargaff rule, they are obey. But if this is a single stranded DNA is single stranded, or when they are replicates, they break down, two base pairs are break down. So in this condition, and when the RNA they do not satisfy this rule, then the function of DNA, they carry genetic information from one generation to others. They help in replication, synthesis of protein, synthesis of all enzymes, and they are very regulates all kind of metabolism. They helps in storage of genetic information, helps in mutation, variation, and evaluation. And then the types of the DNA: A, B, Z, C, D, E. This type of DNA in details I will discuss in the next video. So please subscribe my channel to watch this next video. And if this video is helpful to you, also share it with your friends. I will discuss the what is the definition of it, what is its overall structure, what is its component, who discovered it, and what is the basic structure of DNA. So I think this video is helpful to you. So if this video is helpful to you, please like, comment, and subscribe my channel, and also share it with your friends. Thank you everyone.